Good morning, good afternoon, and good evening to everyone who joins us from around the world. We're so glad you're here. This is Andrew and Renee. Welcome to the Stack series.
It's wonderful to be here. A fascinating topic today.
Today we are exploring the science of caloric restriction mimetics, metabolic switching, and the quest to slow biological aging.
It's a massive topic, really. We're essentially asking a question that sounds like it's straight out of science fiction.
Wait, yes.
Can we get all the biological benefits of scarcity, of, fasting, without the actual misery of starvation? Can we have our cake, eat it too, and still somehow get the longevity boost of an empty plate?
That really is the dream, isn't it? But before we jump into the how, the pills, and the protocols, I think we need to ground this conversation in the why. Because we aren't talking about immortality in the, vampire sense of the word.
No, not at all. That's a common misconception. The goal has shifted dramatically in the scientific community over the last couple of decades.
It's not just about adding years to life, but life to years.
Precisely. The metric that really matters now is health span. Lifespan is, well, it's just the total number of years you're breathing. Health span is the number of years you spend in good health, mobile, cognitively sharp, free from the chronic diseases of aging.
So it's about not spending the last 20 or 30 years of a long life in a state of decline, hooked up to machines.
Exactly. The ultimate goal of geroscience, that's the biology of aging, is what they call squaring the curve.
Squaring the curve, what does that look like?
Imagine a graph of your health over your lifetime. For most people, it's a slow, steady decline. It starts to drop in your 40s or 50s and just keeps going down. Squaring the curve means you maintain a very high level of health and function for as long as possible, say into your 90s, and then you have a very, very rapid decline at the very end.
Right, the rectangularization of the survival curve. You're hiking a mountain on your 95th birthday, you go to sleep that night, you just don't wake up.
That's the ideal. No decade-long battle with multiple diseases. But to even have a shot at that, we have to confront this core paradox that has been at the heart of aging research since the 1930s.
And that would be the caloric restriction effect.
It is, without a doubt, the single most robust, most replicated finding in the entire history of aging science. It started with a researcher named Clive McKay at Cornell almost a century ago now.
What did he find?
He found that if you take an organism, and this has been shown again and again in everything from yeast to worms to fruit flies to rats and even in primates and rhesus monkeys, if you restrict their total calorie intake by about 20 to 40%, while still giving them all the essential vitamins and minerals.
So they're malnourished in calories, but not in nutrients.
Exactly. When you do that, they live drastically longer. We're not talking about a small percentage. In some studies, it's a 30, even 40% extension in maximum lifespan.
That number is just staggering. If you just do the simple math and apply that to a human lifespan, we'd be looking at people routinely living to be 120, 130.
Easily.
But here's the massive catch, the rub. Starvation is miserable.
It's torture. It goes against every biological and psychological drive we have. I mean, think about it.
I get irritable if my lunch is an hour late. The thought of living my entire adult life on a 40% calorie deficit, it sounds like a very long, very cold, very unhappy existence.
And it's evolutionarily nonsensical for us to want to do that. Our bodies are exquisitely designed to seek out and consume energy, not to chronically refuse it. So what we're left with is this proven, powerful intervention that is practically impossible for 99.9% of the human population to actually sustain.
Which brings us then to the central mission of our whole exploration today. If we won't or can't starve ourselves, can we trick our bodies into thinking we are?
Can we find a molecule, a drug, a supplement that flips the same metabolic switches that fasting does?
That is the promise of caloric restriction mimetics, or CRMs. And to really get our heads around them, we have to move past the surface level stuff, the anti-aging creams, and look at the actual machinery of the cell.
The fundamental wiring.
Let's get into that machinery. We'll start with part one, foundations, what we think we know. In the research, I kept coming across this analogy of the body as a factory, and I found it incredibly helpful for visualizing what's really going on.
It is the best mental model by far. So imagine your body, or even a single cell, is a big manufacturing factory. When resources are abundant, when you're eating 3 square meals a day, plus snacks, plus a late-night dessert, what's happening?
Raw materials are just flooding the loading dock.
Constantly. You've got glucose, you've got amino acids from protein, fatty acids. The supply chain is completely full. It's overflowing.
So factory manager sees all this stuff coming in and says, full speed ahead.
Exactly. The factory runs 24/7. It's in growth mode. It's churning out new products, building new proteins, cells are dividing, everything is expanding. But what happens to a factory that runs nonstop without ever pausing for maintenance?
It breaks down, the machinery wears out, gears grind down, belts snap, and trash starts piling up in the corners because the cleanup crew is too busy working the assembly line.
That's it, precisely. You accumulate damage. In the cell, this trash looks like misfolded proteins, damaged mitochondria, the power plants, and just general metabolic waste. This slow accumulation of damage is, at a very fundamental level, what aging is.
Okay, so that's the state of abundance. Now, what happens if you cut the supply line? You stop eating,
Yeah.
you fast.
The loading dock is empty, the factory goes quiet. The factory manager looks around and says, all right, team, we have no new raw materials coming in. We can't build new product. We can't just sit here and do nothing. So let's use this downtime to fix the machines.
And that's the switch, the critical pivot from production to repair.
That is the absolute key to longevity. And what's amazing is we've actually identified the molecular managers who control this switch. The manager in charge of growth of production is a protein complex called MTO.
MTO, that stands for mechanistic target of rapamycin.
Correct. You can think of MTOR as the general contractor on a construction site. Its job is to sense abundance. Specifically, it's very sensitive to amino acids, the building blocks of protein. When you eat a steak, MTOR screams, build!
So it's the gas pedal.
It is the gas pedal for growth. It pushes the cell to grow, to divide, and it actively inhibits the breakdown of old parts because it wants to keep all hands on deck for production.
But the problem is, if that general contractor is screaming, build, for 50 years straight without a break.
You get a factory full of old, broken, junky machinery, and that unchecked growth signal is also implicated in things like cancer and the accumulation of senescent cells. It's growth at all costs.
So who's the opposing manager? Who's in charge of the repair crew?
That would be AMPK.
Adenosine monophosphate activated protein kinase. That's a mouthful. We'll stick with AMPK.
Yes, let's. So if MTR is the growth sensor, AMPK is the low energy fuel gauge. It's constantly monitoring the ratio of ATP, which is the cell's main energy currency, to AMP, which is basically spent fuel.
Like the difference between a fully charged battery and a dead one.
Perfect analogy. When you're fasting or exercising, you're using up your ATP and the levels of AMP rise. AMPK detects this shift and it wakes up.
And when that fuel gauge hits empty, AMPK takes over from MTR.
Immediately. Its first job is to shut down MTR. It basically walks over to the general contractor and says, stop all new construction. We are running on fumes. We need to conserve energy. And then it initiates a critical process called autophagy.
Autophagy. This literally means self-eating.
It sounds a little gruesome, doesn't it? But it's actually one of the most elegant and essential processes in the body. The cell literally begins to hunt down its own damaged components.
Like those misfolded proteins and worn-out mitochondria we mentioned.
Exactly. It finds that junk, wraps it up in a special double-membraned bubble called an autophagosome, and then fuses that with a lysosome, which is like the cell's stomach, full of digestive enzymes.
And it just digests its own trash.
It digests its own trash and recycles the raw materials. It breaks down those damaged proteins into fresh amino acids that can be used to build new functional proteins, or it burns them for immediate energy to keep the cell alive.
So back to our factory. This is the cleanup crew going around collecting all the scrap metal and broken parts from the factory floor and melting them down in the furnace to keep the lights on.
And in doing so, they leave the factory floor spotlessly clean. This deep cleaning is vital. Without robust autophagy, our cells, especially long-lived ones like neurons in the brain, effectively drown in their own waste over time.
This is heavily linked to neurodegenerative diseases like Alzheimer's and Parkinson's.
So to put it all together, the grand strategy of a caloric restriction mimetic is to find a way to artificially suppress MTOR and activate AMPK, thereby triggering this autophagy cleanup process, all while we're still eating normally. That is the core mechanism in a nutshell. But it's not just about these internal energy sensors. The body is a whole system. We also have to talk about the hormonal signals that orchestrate this whole response. And the main one here is ghrelin.
Ah, ghrelin, the hunger hormone. This is the one that usually gets a really bad rap. It's the reason I find myself staring into the refrigerator at 11 p.m.
It's often vilified, you're right. Because in the context of our modern food environment, high chronic levels of ghrelin are associated with overeating and obesity. But in the context of longevity and fasting, ghrelin is actually a hero.
How so? Where does it come from?
It's secreted primarily by specialized cells in the lining of your stomach when it's empty. It then travels through the bloodstream up to your brain, to the hypothalamus, to send the powerful signal, find food now.
But the sources we looked at pointed out something I had never realized about the name itself. The root of the word ghrelin.
Yes, this is fascinating. It comes from the Proto-Indo-European root grer, which means to grow.
Which seems like a total contradiction. We just spent 10 minutes talking about how we need to stop growth to activate repair. Why on earth is a hormone that spikes during fasting named to grow?
It's a fantastic question. The name comes from its potent ability to stimulate the release of growth hormone from the pituitary gland. But more importantly for this conversation, ghrelin is profoundly neuroprotective. Just think about this from an evolutionary perspective. If you are a hunter-gatherer and you haven't eaten in 2 days, what does your brain need to do?
It needs to be sharper than ever. It has to be laser focused on finding a food source, on tracking prey, on remembering where those berry bushes were.
Exactly. If starvation made you cognitively slow, lethargic, and foggy, our species would have died out. Ghrelin is the solution. When it spikes, it actually ramps up cognitive function. It's been shown to stimulate neurogenesis, the creation of brand new brain cells, particularly in the hippocampus, which is crucial for memory. And it's a powerful anti-inflammatory in the brain.
So it's keeping the pilot sharp while the plane is running low on fuel.
That's the perfect way to put it. This completely reframes the sensation of hunger. That gnawing feeling in your stomach isn't just a nuisance to be silenced with a snack. It's a biological signal that your brain is being bathed in a potent neuroprotective and cognition-enhancing cocktail.
That's a huge psychological shift. But I remember reading, there's a bit of a nuance here, that not all ghrelin is the same.
Yes, this is where it gets a little more complex. Ghrelin comes in 2 main forms, acylated and unacylated.
Acylated, that means it has a fatty acid chain attached to it, right?
Correct. A specific enzyme in the stomach, called GO8, attaches an octanoic acid to it. This acylated ghrelin is the active form that can cross the blood-brain barrier to stimulate appetite and growth hormone release. For a long time, the unacylated form, which is actually far more abundant in our blood, was thought to be just inactive biological junk.
And now we know that's not the case.
Now we're learning that unacylated ghrelin may have its own distinct and sometimes opposing metabolic effects. It might be involved in improving insulin sensitivity in some tissues. It's a very complex tango, as some researchers call it, between these 2 forms, and also with leptin, the hormone that signals fullness.
The satiety hormone.
Right.
Yeah.
But for our purposes today, the key takeaway is that the spike in ghrelin during a fast isn't a bug in the system. It's a critical feature designed to protect and enhance your most important organ, your brain, when it needs it most.
Okay, before we move on to the actual drugs and supplements, there's one more foundational piece of the puzzle we have to put in place. The guardian genes. The sirtuins.
Ah, yes. They're sirtuins. You can think of them, especially SIRT1 and SIRT3, as the master supervisors of this entire repair operation. They're a family of proteins that regulate everything from DNA repair to inflammation to mitochondrial health, but they have a very strict requirement to do their job. They are what we call NAD plus attendant deacetylases.
Okay, you're going to have to explain that one for me. NAD plus E dependent.
It just means they run on a specific type of fuel. And that fuel is a crucial molecule called NAD plus O, or nicotinamide adenine dinucleotide. Think of NAD plus as the electricity that powers the factory's entire maintenance department. When you are young or when you are in a fasted or energy-deprived state, NAD plus levels are high. And when NAD plus is high, the sirtuins are switched on.
And what do they do when they're switched on? What's their job?
They get to work. SRT1 is primarily found in the nucleus of the cell, and its main job is to protect your DNA. It helps stabilize the genome, it directs DNA repair crews to sites of damage, and it powerfully controls inflammation. SRT3 is the mitochondrial guardian. It lives inside the mitochondria and protects those power plants from oxidative stress and damage.
So they're not the repair crew themselves, but they're the managers of the maintenance crew. They're the ones with the clipboards telling AMPK and the autophagy machinery where to go and what to fix.
That's a great way to think about it. But here is the central tragedy of the aging process. NAD plus levels plummet as we get older. It's a well-established fact. By the time you reach middle age, you might have less than half the NAD plus levels you had in your 20s.
So the fuel tank for the maintenance department runs dry.
It runs dry. The sirtuins start to go quiet, they can't do their job, and the whole repair and maintenance operation grinds to a halt. And that's when the damage really starts to accumulate and stick.
Okay. So we have the board set. We have the core players, the energy sensors, MTOR and AMPK. We have the recycling crew, autophagy. We have the hormonal hunger signal, ghrelin. And we have the supervisors, the sirtuins, with their NAD plus fuel.
That's the foundational framework.
And now we can get into the really interesting part.
Yeah.
Part 2, the active debate. This is where science disagrees. Can we actually manipulate this incredibly complex, interconnected system with a pill?
And this is where the science gets really exciting and also very contentious. We have several major candidates for a magic pill CRM.
Let's start with the one that is by far the most mainstream. Metformin. Millions of people have this in their medicine cabinets right now for type 2 diabetes.
Metformin is, in many ways, the OG of the longevity field. It's a cheap, incredibly safe drug derived from a compound found in the French lilac flower. And it's been used for decades to treat type 2 diabetes because it's very effective at lowering blood sugar. But the reason biohackers and longevity researchers are so interested in it has nothing to do with sugar directly. It's because of how it works.
And how it works is it's a mild mitochondrial toxin, correct? Yeah. Which on the surface sounds like a very bad thing.
It sounds completely counterintuitive, doesn't it? Take this toxin to live longer. But metformin is a weak inhibitor of something called complex I in the mitochondrial electron transport chain.
So it goes into the cell's power plants and just gums up the gears a little bit.
Just a little bit. It's like putting a tiny bit of friction on the assembly line. This reduces the overall efficiency of ATP production. The cell suddenly isn't making energy as well as it was a moment ago. It notices this drop in energy, this rise in AMP, and what does it do?
It panics and triggers AMPK.
Exactly. It tricks the cell into thinking it's in a state of energy deficit, even if you just ate a carbohydrate-rich meal. And that AMPK activation kicks off all those beneficial repair and cleanup pathways. This is the mechanism that has led to the landmark TAME trial.
TAME, which stands for Targeting Aging with Metformin. This trial is a huge deal, not just for the drug itself, but for the regulatory precedent it could set.
It's absolutely historic because right now the FDA and other regulatory bodies do not recognize aging as a disease that you can treat. You can get a drug approved to treat diabetes or cancer or heart disease, but not aging.
You treat the symptoms of aging, not the root cause.
Right. The TAME trial is the first of its kind designed to prove that by targeting the fundamental biology of aging with a drug like metformin, you can delay the onset of all of these age-related diseases simultaneously. If it succeeds, it could open the floodgates for developing and approving drugs for aging as a primary indication.
But, and this is a very big but that came up again and again in the sources, is metformin actually a good idea for healthy, metabolically fit people?
That is the million-dollar question and the heart of the debate. For someone who is diabetic, pre-diabetic, or even just has poor metabolic health, the evidence is pretty clear that metformin is beneficial. But for a fit, healthy person who exercises regularly, the data is much murkier, and some of it is concerning.
Why? What's the concern?
The concern is that it might actually blunt the beneficial adaptations of exercise.
This is the hormesis problem, right? The idea that a little stress is good. When I go to the gym and lift weights, I am intentionally stressing my muscles and my mitochondria so that they are forced to adapt and grow back stronger.
Precisely. But if you take metformin before that workout, you are adding an artificial stress on top of the exercise stress by inhibiting mitochondrial function. And you're suppressing the very mitochondrial respiration that you were trying to improve with your training.
So you're pressing the brake and the gas at the same time.
In a way, yes. And some studies have actually shown this. Groups of people on an exercise program who also took metformin did not see the same improvements in their VO2 max, which is a key marker of cardiovascular fitness, or the same gains in muscle mass as the groups who just did the exercise alone.
So it seems like it might raise the floor for people who are unhealthy, but it could potentially lower the ceiling for people who are already fit and active.
That is the current consensus among many experts in the field. It's probably not a universal preventative for everyone. It's a therapeutic tool for metabolic dysfunction.
Okay, let's move on to the real heavy hitter in the lab. The one that gets the researchers the most excited, rapamycin.
Yes. Rapamycin is the undisputed gold standard for lifespan extension in animal models. It was discovered in a soil sample from Easter Island, which is known as Rapa Nui to its native people, hence the name.
And it wasn't discovered as an aging drug initially?
No, not at all. It was first identified as an antifungal agent. Then researchers discovered it was a powerful immunosuppressant, and it's been used for decades to prevent organ rejection in transplant patients.
Now, immunosuppressant is a very scary word if you're not getting a new kidney. For most of us, that word means you're going to get sick from common infections and potentially die.
And that is the absolute core of the safety concern with rapamycin. It works by being the most potent known inhibitor of MTOR. It physically binds to a protein and wedges itself into the MTOR complex, shutting it down completely. In mice, flies, yeast, you name it, this reliably and robustly extends life, more so than any other compound tested.
But we are not mice living in a sterile plastic box in a lab. We live in the real world, exposed to viruses and bacteria every single day. If we shut down MTOR, which is critical for immune cell proliferation, don't we shut down our immune response?
In the high daily doses that transplant patients take, absolutely. You would be dangerously immunocompromised and prone to all sorts of infections, but that's not how the longevity field is approaching it. The real excitement is around pulsed or intermittent dosing.
What does that mean?
It means taking a much smaller dose, maybe just once a week or once every 2 weeks. The idea is that you inhibit MTOR just long enough, maybe for a day or two, to get a big pulse of autophagy and cellular cleanup.
But then you allow it to recover for the rest of the week so your immune system can come back online and function normally.
Exactly. You get the benefit without the chronic downside. But it is a very fine line to walk. It's a tightrope. If you get the dose or the timing wrong, you can still experience side effects like mouth sores, poor wound healing, and paradoxically, if you inhibit it for too long, it can actually cause metabolic problems like insulin resistance.
Why is that?
Because it turns out MTOR exists in two different complexes, MTORC1 and MTORC2. You want to inhibit MTORC1 for longevity benefits, but chronic rapamycin use can start to bleed over and inhibit MTORC2, which is actually essential for proper insulin signaling.
So this is not something to be played with lightly. It's a sledgehammer, not a scalpel.
It is a very powerful tool that requires a lot of respect and, frankly, a lot more human research.
Let's pivot to the natural stuff. The supplements you can go out and buy today. For years, the big one in the media was Resveratrol.
The famous red wine molecule. This was championed very heavily by researchers like David Sinclair. The theory was that it was a direct activator of SIRT1, that guardian gene we talked about.
And what's the reality turned out to be?
Largely disappointing, to be honest. The initial data in simple organisms like yeast was spectacular, but when it moved into mammals, the results were very inconsistent and hard to reproduce. The main hang-up is bioavailability.
Meaning our bodies just can't absorb it well.
Exactly. You can drink a glass of red wine or pop a resveratrol pill, but your liver sees it as a foreign compound and metabolizes it almost instantly. Very, very little of the active compound ever reaches the cells in your body where it would need to work.
So you'd have to drink an absurd amount of wine.
You'd need to drink hundreds of bottles a day to get anywhere near the therapeutic dose that was used in some of the mice studies. And that would obviously cause other more immediate problems for your health. So it's largely considered to be overhyped at this point, although some researchers still believe it has potential if formulated in a way that protects it from the liver.
What about a more direct approach? Instead of trying to activate the sirtuins, what about just giving them more fuel? This is the idea behind NAD plus precursors like NMN and NR.
This is a much more promising avenue, in my opinion, but it's still hotly debated. The logic is simple. We know NAD plus levels decline with age, and sirtuins need NAD plus to function. We can't just take an NAD plus pill because the molecule is too big to get into our cells. So the strategy is to take the building blocks.
NMN, which is nicotinamide mononucleotide, and NR, nicotinamide riboside.
Correct. The body can absorb these smaller molecules and then use them to synthesize new NAD+ inside the cells.
The debate here seems to be less about the theory and more about the logistics and efficacy.
Exactly. First, there's the gut microbiome. Does the expensive NMN powder you swallow actually make it into your bloodstream, or do the bacteria in your gut just have a feast and eat it all first? And second, is the compound even stable? There's evidence that NMN can degrade fairly quickly at room temperature if not stored properly.
So you might be paying a lot of money for a supplement that is turned into useless powder by the time it gets from the factory to your shelf.
It's a classic case of the potential for expensive urine. That said, some of the more recent human clinical trials are showing that certain formulations of NR and NMN can successfully raise NAD+ levels in the blood. The big unanswered question is whether that increase in the blood translates to increased levels inside the tissues that matter, and ultimately whether that translates to a longer, healthier life in humans. We just don't know yet.
There was one more natural compound that really caught my eye, mainly because of the interesting dietary sources that were listed. Spermidine.
Yes. Named for obvious reasons, as it was first isolated from semen. But thankfully for supplement manufacturers, it's also found in things like wheat germs, soybeans, aged cheese, and mushrooms.
We'll focus on the wheat germ, I think.
Good idea. Spermidine is fascinating because it seems to work differently from the others. It appears to bypass the mTOR and AMPK sensors and induce autophagy directly through a different pathway. It's a type of molecule called a polyamine, and it plays a key role in stabilizing DNA and promoting the cellular cleaning process.
And the evidence for this one is actually pretty good.
The epidemiological evidence is quite strong. There are studies showing that human populations who consume diets high in spermidine, people in the blue zones, for example, tend to have greater longevity. It's also been linked to better cognitive health and aging. The main challenge, as with many of these things, is getting a high enough dose from food alone. You'd have to eat a lot of aged cheese.
It was something like wheat germ. The sources mentioned that the oils in it go rancid very quickly, so the quality and processing of the supplement really matters.
It matters a great deal. You need to use defatted wheat germ to get a stable, potent product.
So we have this medicine cabinet full of potential cheats, metformin, rapamycin, NMN, spermidine. But what the sources really started to suggest is that the real breakthrough isn't finding a single magic pill. It's about a new strategy, a new way of thinking. This brings us to part 3. Frontiers, what might change everything. And it's this concept of moving from chronic intervention to cyclical.
This, I think, is the most important conceptual leap for anyone interested in this topic. For a very long time, the thinking in the field was very binary. It was, mTOR is bad, AMPK is good, growth is aging, repair is longevity.
So the logical conclusion from that was, find a way to crush mTOR forever, and you'll live forever.
But biology is never that simple. It's about balance. If you crush mTOR forever with a chronic high dose of rapamycin, or you starve yourself forever with extreme caloric restriction, you don't become a superhuman. You become frail.
What does that look like?
You can't build or maintain muscle, so you develop sarcopenia, or age-related muscle wasting. Your immune system, which needs mTOR to build new immune cells, collapses. Your skin thins, your hair falls out, wounds don't heal.
So you might technically live for a long time, but you'll be a stiff wind away from breaking a hip and dying from pneumonia.
Exactly. You become a fragile, long-lived creature. The realization now is that we need both signals. We need to mimic the ancestral rhythm of our environment. We evolved in a world of feast and famine.
So the goal isn't to pick a side, but to engineer that rhythm into our modern lives. The pulse theory.
That's the frontier. Think of it in 2 phases. Phase 1 is the cleanup or catabolic phase. This is when you trigger repair. You fast, or you use a mimetic for a short period, a pulse of rapamycin, maybe some berberine or metformin. You activate AMPK, you suppress mTOR, and you induce a powerful wave of autophagy. You scour the body for junk.
That's demo day on the renovation project, tearing out all the old rotten stuff.
Perfect analogy. But you can't live on a demolition site forever. You have to rebuild. That's phase 2, the rebuild or anabolic phase. You break your fast. You eat a high-protein meal. You go to the gym and lift heavy weights. You intentionally spike mTOR.
And that's construction day. You use those clean, recycled materials to build new, strong muscle, to replenish your immune cells, to repair your mitochondria.
The real magic is in the cycling back and forth between these 2 states. It's about developing metabolic flexibility, the ability of your body to gracefully and efficiently snap between burning fuel for growth and hunkering down for repair.
And this framework explains why someone like David Sinclair's personal protocol, regardless of the controversy around the individual components, looks the way it does. It's a stack of different compounds, but the underlying philosophy is often this kind of rhythmic application.
Yes. And we're starting to see new agents being explored specifically for their role in this cyclical strategy. Take glucosamine, for example.
The supplement for joint pain.
That's the one. But it turns out it may do a lot more than just lubricate your knees. Some fascinating research suggests it acts as a caloric restriction mimetic by partially blocking glycolysis, the process of burning sugar for fuel.
So it forces the cell to switch to burning fat and protein for energy, which mimics a low-carb or fasted state.
And that state, as we know, triggers autophagy. It's a gentle, hormetic stressor.
That's a massive rebrand for glucosamine. And then there is the senolytics. the zombie cell hunters.
This is the ultimate cyclical hit-and-run strategy. So, senescent cells are cells that have accumulated so much damage they should die, but they don't. They enter this zombie-like state where they stop dividing, but they refuse to undergo programmed cell death.
And they just sit there, causing trouble.
They're terrible neighbors. They sit in your tissues and spew out a cocktail of inflammatory chemicals. It's called the SASP. And this inflammation can actually turn the healthy cells around them into zombies, too. They are the rotten apples that spoil the whole barrel.
So what do senolytics do?
Senolytics are a class of compounds. Some are drugs, some are natural, like fisetin from strawberries or quercetin from onions, that are specifically designed to selectively kill these zombie cells. But you absolutely do not take them every day.
It's a pulse strategy.
The most extreme pulse strategy. The idea is to take a very high dose for just 2 or 3 days in a row, once every few months. You go in, you nuke the zombie cells, clear them out, and then you stop completely to allow the healthy tissue to heal and regenerate in that cleared out space.
It's purely cyclical. Clearance, then regrowth.
It has to be.
But this all begs a huge question. How on earth do we know if any of this is actually working for us as individuals? We can't just take rapamycin for 10 years and wait to see if we outlive our friends. We need feedback in the short term.
We need biomarkers. This is the whole concept of precision longevity.
Yeah.
We need surrogate markers that can tell us if we're moving the needle on our biological age. And this is where tools like the Horvath clock come in.
Epigenetic clocks.
Yes!
Yeah.
We can now take a blood or saliva sample and look at the methylation patterns on your DNA. These are little chemical tags that get added to or removed from your genes over your lifetime in a predictable way. By analyzing these patterns, we can calculate your biological age and compare it to your chronological age from your birth certificate.
So I might be 40 years old chronologically, but my DNA methylation pattern looks more like that of a healthy 35-year-old.
Or a 55-year-old, if you've been burning the candle at both ends. We can then combine this epigenetic data with real-time data from wearables, like continuous glucose monitors or CGMs, to see how our diet is affecting our blood sugar and insulin spikes. And then we layer on traditional blood panels looking at markers of inflammation like C-reactive protein or lipids like ApoB.
So the future isn't a one-size-fits-all pill. It's an AI-driven system that looks at my personal data stream, my epigenetics, my glucose, my inflammation, and says, okay, you've been in growth mode a little too long this month, your biological age is ticking up, it's time for a 3-day fast and a pulse of a senolytic.
That is exactly the future. A completely personalized, data-driven approach to customizing your own cleanup and rebuild cycle. It moves longevity from the realm of hopeful guessing to the realm of personalized engineering.
Let's zoom out now for our final section, part 4, the implications. If this works, if we can actually use these strategies to meaningfully extend health span and compress morbidity, what does that do to society?
It changes everything. It fundamentally redefines what it means to be human. The first, and maybe most practical argument is economic. It's called the longevity dividend.
What's that?
The idea is that if we can delay the onset of aging by just a handful of years, say 7 years, we would save trillions upon trillions of dollars in healthcare costs, because we would no longer be spending the last decades of people's lives treating a cascade of chronic, expensive, age-related diseases.
We stop playing this endless game of whack-a-mole with heart disease, then cancer, then Alzheimer's, and we treat the single root cause of all of them, which is the aging process itself.
Precisely. But then you get into the much heavier ethical questions. These are sometimes called the Mackey objections, and the first one is inequality.
Will this create a biological caste system, a world of the long-lived, where the wealthy can afford these treatments and live to be 150 looking like they're 40, while the poor are left to age and die on the natural timeline?
It is a deeply valid and terrifying fear. If the most effective protocols are patented drugs that cost thousands of dollars a month, or complex stacks of expensive supplements, then absolutely, access will be profoundly unequal, at least at first. The counter-argument, the more optimistic view, is that technology tends to democratize over time. Mobile phones were once multi-thousand-dollar toys for billionaires. Now they're ubiquitous.
Then there's the overpopulation argument. If people stop dying at 70 or 80, won't we just run out of space and resources?
Another common objection. The counterpoint here is that in most developed nations where these technologies would first become available, birth rates are already at or below replacement level. The problem isn't too many people, it's that the population is getting older and less productive. A healthier, longer-lived, older population could remain productive and contribute to society for much longer.
What about the psychological side? The idea of ennui or boredom. Will life lose its meaning and urgency if it's too long?
It's a philosophical question, really. Does the deadline of death give life its value? Perhaps for some. But the counter-argument is that healthy, happy people generally don't want to die. The drive to learn, to create, to explore, to connect with loved ones, that doesn't seem to have a natural expiration date as long as health is maintained.
It seems like there's a big psychological shift that has to happen too. The sources talked about this idea of the warranty period.
This is a powerful way to reframe our relationship with our own biology. From a purely evolutionary standpoint, nature only really cares about you until you've successfully reproduced and perhaps raised your offspring to the point where they can survive on their own. Once you're past, say, 40 or 50, you're in the post-warranty period.
Nature has no plan for us after 50.
It has no plan. You are biologically drifting. And that can sound depressing, but I find it incredibly empowering. It means that the second half of life is a blank slate. It's an engineering challenge. It is up to us to design it, to maintain the machine. We are not fighting nature's grand plan by trying to live longer. We are stepping in where nature's plan simply ended.
So as we wrap up here, what is the single biggest takeaway for you listening right now? We can't prescribe drugs or give medical advice, obviously, but the core principles seem very clear.
The principles are timeless and simple. The first is, do not eat all the time. Our bodies were not designed for the constant 24/7 abundance of the modern world. Introduce periods of scarcity.
I read a great line once: comfort is a signal for decay.
That is the perfect mantra for this entire conversation. If you are always full, always at a comfortable temperature, and always sedentary, your body is receiving a constant signal that the environment is easy. It doesn't need to be resilient. It doesn't need to waste energy maintaining its repair machinery.
So you have to introduce hormesis.
Yeah.
A little bit of good stress.
A little bit of stress is the signal for life, whether that's the metabolic stress of a fast, the thermal stress of a cold plunge or a sauna, or the physical stress of a hard workout. You have to periodically wake up those factory managers, AMPK and the sirtuins.
You have to remind them that the environment is still challenging, so they better keep the machine in top working order.
Precisely. The science will continue to evolve, new molecules will be discovered, but the basic principles of this rhythm of stress and recovery, of cleanup and rebuilding, are timeless.
A perfect place to leave it. We are, in the end, the architects of our own biology.
Indeed we are.
Before we wrap up, thank you to all our partners, sponsors, and scholars who made this podcast and the research possible. We're truly grateful for your support. And to our listeners, thank you for being part of this journey with us. If you enjoyed today's episode, be sure to subscribe and follow Stack Studios for more informative content, and support us on Patreon. Until next time, stay curious, keep learning, and let the search for wisdom guide you forward. Have a great day.